We were always disinfecting our gloves, and I was curious how effective that process is to directly apply that disinfectant to our gloved hands when we're directly handling mice.
Welcome to The Last Word, the official JAALAS podcast dedicated to connecting and informing the laboratory animal science community. Each episode takes a closer look at recent articles published in our journals, JAALAS and LAS Pro, and features interviews with professionals advancing the field. I'm Jennifer Smith, your host. As a laboratory animal veterinarian and the Director of Education and Scientific Affairs at JAALAS, I look forward to sharing these conversations with you. Let's discover something new together. With me today is Dr. Julia P. Smachlo. Julia is a clinical veterinarian at Biomere in Worcester, Massachusetts. She recently completed her laboratory animal medicine residency at the University of Minnesota, where she also completed her DVM degree. Julia also holds a Master of Public Health degree, which she also obtained from the University of Minnesota School of Public Health. We are thrilled to welcome Julia today and look forward to hearing about her recent JAALAS paper titled Evaluation of the Efficacy of Disinfectants in Reducing Contamination of Bacteria and Viral Murine Pathogens on Gloved Hands. Wow. Julia, thank you so much for being here with us. Thanks for putting up with that long intro. We would love to hear more about yourself, your research background, the team you worked with, and really just how you got interested in this great subject for the paper.
Absolutely. So thank you so much for having me on the podcast today. I'm very excited to be here and chat about my paper. As Jennifer stated, I went to vet school at Minnesota, and I went into vet school initially wanting to do veterinary public health. So Minnesota has a great dual degree program for that. I wasn't aware of laboratory animal medicine in the veterinary capacity going into vet school. So Minnesota exposed me to that, and that then became my new route as a veterinarian.
Oh, that's so great, Julia. We love hearing about the background of our guests. That's probably the most fun part of this podcast. And we love hearing how people get into lab animal. And especially for those of you guests who are just starting out, we want you on this podcast and we love hearing about your start to getting into this industry. So that's wonderful.
Yeah, so laboratory animal medicine is kind of a great field because you get to work not only on a population level, but also still work on an individual health level. And for me, having an interest kind of in operating and working in that One Health bubble, working with animals for the benefit of human health as well, laboratory animal medicine absolutely checked all my boxes and has been a very fulfilling career so far for me. On to more my paper. So the rest of the team on my paper. So listed as the second author of my paper is Dr. Marcia Hart. She was very instrumental in helping direct the study design of this paper, including selecting some of our more prevalent pathogens that we might want to look into, and also the inclusion of our genomic copy numbers for evaluation to help quantify the infectious material detected via PCR. Michael Evans was the statistician who was also incredibly helpful for guiding and performing the statistical analysis portion of this project. And Dr. Brenda Kick was my faculty mentor at the University of Minnesota for this project. This project was completed as part of a residency research project in fulfillment to get first author papership for ACLAM eligibility. And Brenda was incredibly helpful in her expertise and guidance for the development of this project, implementation, and putting together this paper.
Oh, that's fantastic. We really love it when people give a good shout out to their team, and it does take a team. And especially we've worked with some of those folks already on some of our committees. So we love hearing their names again and just hearing in their day jobs what great work they do. So that is fantastic. Well, one thing your team did really well was write a fantastic introduction. And we would love to hear just more about it. I love hearing about those pesky infectious agents that I know you're going to tell us about, cleaning, the monitoring methods, just all these things that we consider in our lab animal program. So you really hit hard on that in the intro. So we'd love for you to summarize some of that info for us.
Absolutely. So as all of us know, working within our animal research facilities, biosecurity practices are really critical for not only ensuring the health of our animals that we're working with, but also for ensuring the validity of our research and trying to minimize any other potential confounding variables. For our mice, say the vast majority of all laboratory animal mice are specific pathogen free. So we exclude a lot of agents because we know that either they affect the health of the rodents and or they also are potential confounding research variables. We have many practices in place for helping to maintain the health of our rodents in our laboratory animal colonies. And one of the biggest and most commonly used ones is probably going to be our microisolator health system. So that starts off with our microisolation caging, which helps to really establish that their exposure is within the cage and their greatest risk of encountering anything infectious or any other microbial contamination is going to be when that cage is opened. So depending on your institution or the health status of your mice or different strains that you're working with, you may employ different levels of this microisolator technique. Pretty broadly, this may include only opening the cage of your mice inside a ventilated biosafety cabinet that has been appropriately disinfected, wearing your PPE, disinfecting your gloved hands before handling or working with these mice, possibly having sterilized or disinfected equipment or any other tools that may come into contact with the mice as well. So something that really stood out to me after beginning my residency was we have recommendations for generally trying to change gloves between mice of different strains, immune statuses, possibly different investigators and rooms. And we were always disinfecting our gloves. And I was curious how effective that process is to directly apply that disinfectant to our gloved hands when we're directly handling mice. Do we need to be changing our gloves more frequently? Is this effective for up to 10 cages? Can we do a whole side of a rack? And there wasn't a ton of literature available for me to be able to answer that question. So that was really the antithesis for this research project, was to evaluate how effective the application of some of our commonly used disinfectants were in trying to mitigate the contamination risk of both just general gross microbial contamination when handling multiple cages of mice and specific pathogens. And so for this study, we selected 3 pathogens of interest. The first is Murine norovirus, which is, according to a 2023 study, the most common viral pathogen in laboratory mice, with a reported prevalence rate of up to 20% when via PCR detection. I'll refer to it as MNV moving forward. MNV is transmitted fecal orally, and in our immunocompetent mice, they may be subclinically infected, but have pretty persistent fecal shedding, so possibly still creating greater infectious risk. MNV transmission has been documented in multiple food safety studies as well, where transmission can occur between fomites, such as gloved hands, onto food products. And for any immunocompromised mice who are exposed or infected with MNV, there is a risk of clinical disease, which might include signs like weight loss, diarrhea, and can result in mortality in certain strains. The second agent that we were interested in was Helicobacter species, generally as a group, and these have a reported prevalence rate of up to 14% by fecal PCR testing. This is also primarily transmitted fecal orally, but there have been some reports of dust or fomite transmission in our immunocompetent mice after Helicobacter colonizes the cecum and colon in certain species. This may lead to long-term fecal shedding. A Helicobacter infection can result in clinical disease in susceptible or immunocompromised strains that may include signs such as hepatitis, colitis, and other signs consistent with more of an inflammatory bowel condition such as diarrhea or rectal prolapse. And our third agent of interest was Rodenibacter species, specifically Rodenibacter hylae and Rodenibacter pneumotropicus, with reported prevalence rates by PCR of up to 16 and 10% respectively. Rodenibacter is transmitted via direct contact, and some clinical signs that we may see in susceptible or immunocompromised mice could include conjunctivitis, dermatitis, preputial and orbital abscesses, to name a few. So for our disinfectants of interest, University of Minnesota Research Animal Resources commonly used Rescue, which is a hydrogen peroxide-based oxidizing agent, as the core part of our microisolator technique. and hydrogen peroxide-based oxidizing agents are very commonly used both in laboratory and veterinary settings for general disinfection. So Rescue is a broad spectrum, fast-acting disinfectant. And then our second disinfectant of choice was 70% ethanol, which is a denaturant. It does have a broad efficacy, but it does not have as wide of a spectrum of efficacy as Rescue. During this study, we also employed PCR testing as well as ATP testing. So PCR testing is to detect the genomic material of our pathogens of interest. And ATP testing is a broader microbiological monitoring system that has been fairly routinely used to assess the efficacy of sanitation and disinfection within our research facilities. So the overall goal of our study was to determine if our common laboratory mouth pathogens readily contaminate gloves after handling as detected by PCR. And if they do readily contaminate those gloves, are we able to reduce in quantity or eliminate the genomic material with the application of disinfectants to our gloves during routine handling? Our study hypothesized that the application of Rescue and 70% ethanol to gloved hands before handling mice would be more effective than water or no disinfectant in reducing contamination with MNB, Helicobacter species, and Rhodonobacter species on gloved hands.
Wow. Well, thank you so much for covering all that. There was a lot of material there, and I really appreciate you summarizing that for our listeners. So we'd love to hear about the study design. I know you kind of delved into that a little bit in the intro, but it would be great if you could just set up the study design for us.
Absolutely. So for this study, we sourced 40 cages of immunocompetent mice, all on a C57 Black 6 background, from an established in-house colony with known natural infections, MNB, Helicobacter species, and Rhodonobacter species. We tested at a cage level all of these cages to confirm infection or establish which cage had which pathogen. And we found that 36 of our cages had at least one of these pathogens. And the 4 cages that were not positive for any were removed from the study. For the first part of this study, we conducted a brief pilot study to assess if we could detect pathogen contamination on gloved hands after handling these mice. So all cages were brought into a disinfected biosafety cabinet, one at a time. They were opened inside the cabinet, and 2 mice in each cage were briefly held in a scruffed position. No disinfectant was applied to the gloved hands before handling, and a new pair of gloves was worn for each cage. We received the results of this pilot study before moving on to the larger part of our study to confirm that we did detect these pathogens via PCR on our gloved hands after handling. For the main experimental part of our study, these 36 cages were randomly assigned to one of 4 experimental treatment groups. We had 9 cages of mice in each group. Our first group, mice were only handled with the 1:32 dilution of Rescue. In our second group, mice were handled with 70% ethanol. In our third group, mice were handled with sterile water. In our fourth group, served as a positive control group, and mice were handled without the application of any liquid or disinfectant. For each group, a single pair of gloves was worn to handle these 9 cages consecutively. Each cage was brought into our disinfected biosafety cabinet, opened, disinfectant, applied to our gloved hands before handling the mice. 2 mice in each cage were again held for roughly 15 to 20 seconds in a scruffed position and then returned to their cage. That cage was brought back to the rack. The next cage was selected, brought into our biosafety cabinet, opened up, disinfectant applied. And at that point, instead of handling the mice, we collected an ATP swab of our gloved hands. We repeated this process for 4 cages of mice. And on that 5th cage of mice, we also obtained a PCR swab. We also obtained a PCR swab after handling the 9th cage or our last cage of mice for each treatment group. And then we repeated this entire sampling process 2 additional times.
Wow. Thank you so much for putting all of that together for us. Like I said, it was a robust study design. And we really appreciate you highlighting the pilot study, because I think that's an area that people sort of maybe think is not important, or they maybe don't include it in their manuscript submissions for articles. So we really appreciate you having that background material in there, but also highlighting it so well for the podcast. So thanks. All right. So on to results. I'm dying to know, did it work? Did you confirm your hypothesis? I guess we could kind of start there.
Yeah, so we did not confirm our hypothesis. We did not have statistical evidence that our rescue and ethanol treatment groups had significantly lower quantities of our genomic material of our pathogens of interest. Interestingly enough, we did detect all of our pathogens of interest during, the initial pilot handling study, with Helicobacter collected the most frequently. And for our experimental design, where we were assessing the pathogen contaminations with our different treatment groups, we found that all three of our agents and all treatment groups were still detected via PCR on our gloved hands. Of note, there was only one significant finding, which was that Rhodinobacter species was not detected after handling 9 cages of mice in our water handling group, which was significantly different from our no product group.
Interesting.
Mm-hmm.
Anything in those results? Did anything surprise your group?
Yeah, so we did have our positive control group, our no product group, did have predominantly higher genomic copy numbers than our other treatment groups, with an exception here or there. And we did find that our rescue treatment group for Helicobacter and MNV decreased after handling from the initial PCR values after handling 4 to 9. So over time, we were seeing a reduction. And this was true as well for the water treatment group for all 3 pathogens. There was a decrease in our genomic copy numbers. However, this wasn't statistically significant, just a trend we observed. We were surprised that ethanol, which we expected to have a reduction in our genomic copy numbers, actually had an increase in our mean copy numbers after handling 4 cages up to after handling 9 cages. So this was pretty surprising to us. For our ATP results, we did find that after handling up to 4 cages of mice consecutively, there was a significant difference between our rescue and water group and our ethanol and water group, as well as our ethanol and no product group. And there was a significant difference in our collected ATP reactive light units out to 8 cages consecutively. And for ethanol and water, this also was present for up to 7 cages consecutively. Throughout the 9 cages this was recorded, there was never a point where there was a significant difference between the rescue and ethanol group.
Wow. And those are interesting findings. Some of those really did surprise me. So I was anxious to read in the paper in the discussion section, because your group did a really good job of kind of summarizing some of that, as well as pointing out maybe some of the limitations to the study. We always really appreciate hearing that, or reading that rather, in the article, and then, of course, talking about it on the podcast. So we'd love for you to talk a little bit more about your discussion, any limitations, anything else that you think our listeners would find interesting.
Absolutely. So our results were not 100% supportive of each other, because based on our PCR data, it looks like rescue and water were superior in reducing our quantity of genomic material over time, whereas based on our ATP data, rescue and ethanol were superior to water specifically. So it makes the analysis, and interpretation of our results a little more challenging with how we want to actually be able to apply what we found. But ultimately, to summarize our major findings, we were able to detect genomic material from all 3 of our pathogens on gloved hands after handling mice, and neither rescue nor 70% ethanol was fully effective in eliminating transfer of those pathogens. We did determine that all of those pathogens may carry a risk of contamination on gloves, as detected via PCR after handling our naturally infected mice. And despite the application of rescue, 70% ethanol between and before handling multiple cages of mice, these pathogens were still detected on PCR. So this component of micro-isolation technique may not be as effective as I initially thought going into this project. While the mice in the no product group did have higher mean copy numbers, this wasn't statistically significant. However, it may indicate that there may be a benefit to the application of applying a liquid based on the PCR results to the gloved hands of mice before handling. This was supported by the complementary ATP testing, which found that microbial contamination was reduced with the application of rescue and 70% ethanol, compared with both water and no product for handling up to 3 cages consecutively. So as you already mentioned, Our hypothesis was unfortunately not confirmed. We did not have evidence that there was a difference in the treatment groups in reducing our transfer of our pathogens of interest after handling 4 or 9 cages of mice, with the one exception that we discussed of Rodentobacter. As an outlier of note, we did have fairly unexpectedly high variability between our PCR sampling events, so this may have contributed to our statistical analysis and lack of significant findings. On a broader level, MNV transmission by gloved hands has been well established in our food safety industry, and gloves inoculated with MNV are capable of transmitting infective MNV onto produce, bread, and stainless steel surfaces. Studies like this support our findings that MNV may be transferred via fomites. However, we did not assess whether or not this material remained infective. That was beyond the scope of this study. So we know the genomic material is there, but we do not know whether or not it is capable of being infectious.
Well, thanks for summarizing a lot of that really complicated information for us. We appreciate that. And I really love reading that in the discussion, just some of those finer points, especially about the MNV and the food safety industry. I definitely appreciate that. And I appreciate your tie in there because there's always some other ways to get some of this information, right? And design your study. So I definitely really applaud your group for taking a wide scope at this. All right. Well, we are actually at the end, which is always so sad when the episodes are at the end, because we just love hearing from our guests and we would love to talk to you all day about this paper and more of this. But anyway, we always have one final question for our guests, and here is yours. What advice would you give to lab animal programs that are thinking about changing up their current practices on how they're doing their cage changing, handling, and with all this information now that you know, what kind of advice would you give somebody?
I wouldn't want to draw too many conclusions from this study. This study is definitely a preliminary study. Both of the PCR and ATP modalities that were employed only detect the presence of genomic material, and this material may be alive or dead. So potentially our rescue or 70% ethanol did kill our pathogens of interest, but we can't definitively say. And just because there is a positive detection on PCR, this doesn't correlate to the infectivity of this detected genomic material. So we shouldn't jump to any conclusions that because it's still there, it could necessarily cause infection in the next mouse I handle on my glove. Further studies need to be conducted to be able to more fully elucidate this risk. From a kind of gross contamination standpoint, our ATP findings, we did find that genomic material from these pathogens was detectable on our gloves after handling, and we were not able to fully disinfect our gloves between cages of mice, both from an ATP standpoint as well as from a PCR standpoint for both results. People may want to consider following our refined handling techniques for our rodents, so using something as a tube that can be disinfected fully between each cage of mice if you're doing cage change out. However, institutions should still follow whatever guidelines make sense for their rodent colonies. So whether they establish glove changes based on, strain, the immune status of mice, size of the rack, or investigator possibly, use what makes sense for them. Further work really should be done before large conclusions or changes of practice are implemented based on this paper.
Excellent. I love that summary. You did a great job of just kind of putting it all together there and giving that nice, hey, here's some information, but make sure it works for your institution. So we love that. So thank you again. And thank you for being here. This has been great. All
Thank
right.
you for asking me.
Yes. Thanks for joining us on this episode of The Last Word. We'd love to hear your thoughts, so be sure to leave your feedback in the comments. And don't forget to subscribe for more great content. We encourage you to submit your research manuscripts to our journals, JAALAS and LAS Pro. We're always looking to feature impactful studies. You can also connect with us on our social media channels or email us directly at info@aalas.org. We appreciate your listening today, and we'll see you next time on The Last Word, an AALAS podcast. Until then, stay curious and stay connected.